So we did exactly the same on a group of subjects, healthy subjects. We took them and we gave them a standard blood glucose tolerance test. And that is where you drink a disgusting amount of glucose, really horrible. That sends your blood glucose up. And then we prick your finger every few minutes and test your blood glucose. It's a super, super simple experiment. So we did the controls first, and we found that their blood glucose peaked at about, around an hour after they drank the glucose. And then in the experimental subjects, we gave them a burst of red light. And then we initiated the experiment, got them to drink the glucose, prick their fingers, and we found that their blood glucose levels just didn't peak in the same way. They were significantly reduced. So we worked their mitochondria really hard. The mitochondria pulled that glucose, that excess glucose, out of your blood. And so we created a situation in which we lowered blood glucose.
So can you give us an idea about the magnitude of that blood glucose decrease, just so that we can get an idea?
It varied between people, but it's around 17%. I mean, that's not marginal. And I would add on to this, we haven't published this yet, but we're 3/4 of the way through a study on diabetic people with type 2 diabetes. We've got exactly the same result, absolutely the same result. So we wanted to pilot it in normal subjects first because it was easier to get the ethics, and we had people all around us. But this is translatable. It is fully translatable. There are a number of other things around this domain that are worth paying attention to. So if you look at epidemiological studies, you find that people who spend much more time in daylight have much better regulation of their blood glucose than people who spend their time in an office environment. Light is key because it's the spectrum of light. So we give red light. We work mitochondria harder. They eat glucose. We can slow mitochondria down as well. So if we give blue light, that slows mitochondria down. And then when we do a blood glucose test, we haven't done this on humans, we've done it on animals. We give them blue light, we give them a blood glucose test, and their blood glucose peaks very high. So light is influencing metabolism systemically. And one of the issues we have in society now is that we're spending a lot of time indoors. We're also spending a lot of time under LED lighting. And LED lighting,
now all these workplaces are now creating these diabetic light environments.
You're not being controversial, you're stating a fact. And they had no vitamin C. Something was taken away from them. And that is very similar to what's happening with light. Someone has taken away the infrared. Scott Zimmerman said someone's robbed it.
Yeah.
And they didn't have a right to do that. And I think that's absolutely right. Something has been taken away from us. Millions of years you've evolved over, you've evolved in sunlight. And it's established our immunity, it's established our metabolism. And then in the late 1990s, the LED was invented, and no one realized the unbelievable effect that potentially could have.
The clinical implication of, or the clinical focus of improving metabolic health in lifestyle medicine is very much focused on diet. It's low carbohydrate, carnivore diet, which is an effective intervention. But there is no talk about light from those quarters. Whereas we haven't mentioned it, but one of the mechanisms by which infrared light is having an beneficial effect, is through the production of melatonin in the mitochondria, and that is the work of Scott Zimmerman and Russell Ryder. And I think that's a paradigm shift in understanding that it's not actually the pineal gland that is pumping out most of the melatonin in the body. It's actually during the day in all your cells on exposure to infrared light.
Yeah, and what a shock that paper was. An absolute shock. It can't be a pineal gland because, as you age, your pineal gland tends to calcify anyway. You use it as a landmark sometimes on an X-ray. So the idea that your mitochondria are packed with melatonin is a game changer. Why is it there? What is it doing? We think it's probably buffering and protecting, but it's in the cell. It's not floating around, and it's not coming from the brain. That is a very, very important paper. It shocked us in the field. I don't think it's sunk in with people really.
I was going to make that exact point, Glenn, which is I don't think people have realized the implication of that paper. It's called Melatonin and the Optics of the Human Body. It's like one of those albums, the music albums that gets released and no one really realizes it, and then 2 decades later everyone starts playing it. That's how important it is, I think.
Dense. Is there lots of mitochondria there, or is it just...
Your photoreceptors in your retina have got more mitochondria than any other cell in your body. Now, your retina burns more energy than any other part of your body. It burns more energy than your muscles. It burns more energy than your heart. It burns more energy than your liver. It is in we talk about it as being on a metabolic cliff. It is in a very dangerous situation because if you burn lots of energy, you generate lots of rubbish, right? So that dustbin is filling up because you're burning lots of energy. The retina is unique in that respect. Retina is incredibly energy dependent. And that made life very easy for me because when I came into this field, I came in as a retinal person. And so my home territory was completely fertile for trying to attack this problem. But Mike has now gone on to do it with type 2 diabetics. And I have seen early signs of that data, and it looks great. Right? So, and with that, we were shining red light on a region of people's backs for about 15 minutes. I think we could do better. I think we can give less light, shorter time periods, and get the same effect. Now, a natural question from this is shining light on a little bit of someone's back, small proportion of the body area. Why are mitochondria all over the place consuming more sugar? And the thing is that if you manipulate mitochondria in one part of the body, the story gets out. Mitochondria are talking to one another right across the body. And we know that from a number of experiments that we done ages ago, where people would manipulate mitochondria in one bit of an animal, and then they'd see them change the following day over here. No one really had a great idea as to what was going on. But of course, you can't have metabolism changing in one place and not the other. So we did find in animal experiments, the moment we hit them with a burst of red light, a signal changes in the blood. And that signal is something called cytokines, and they're very often associated with inflammation, but they're also signaling molecules. So mitochondria are affected by the red light. They change their metabolism, and they leave a message that goes out, and that message goes out to mitochondria in other parts of the body that do not receive the red light, and those mitochondria change and come into line with the ones that were exposed.
Wow.
This is really super important work. And Jay, I think you know this work quite well. This is out of the group in Australia looking at the ability of remote photobiomodulation to have a protective effect, and particularly on the dopaminergic neurons of the substantia nigra. So the toxin MPTP is used routinely as a way of creating a laboratory model of Parkinson's disease. MPTP, discovered by accident in a lab trying to make an illicit drug, gave all these drug users Parkinson's that was completely irreversible because the neurons were just wiped out. So here's the study. Here's an animal that was injected with saline. Here's an animal that's injected with MPTP, and you see that the infrared light, the MPTP has wiped out the substantia nigra neurons. There are all these little brown neurons. So they're gone. Now, here's an animal that was pre-treated with infrared light and then injected with saline. And we see, no difference really from the saline, okay? Here's an animal that was pre-treated with infrared light, then injected with MPTP. And look, there's sparing of the dopaminergic neurons. Now, here's the sticking point. The infrared light was applied to the back of the animal, not to the head, not to the brain, but to their back. And this had a remote protective effect against MPTP toxicity. All right, now we know infrared light. Let's take another example. Rodrigo et al. Put 3 wounds on the dorsum of a rat. And then they treated the wound at the head only. They had 4 groups. One was in the red and infrared laser group. Healing was more advanced in the wound located furthest from the point of laser application. So in other words, they treated here and this wound got better faster. Remote photobiomodulation.
Other people in other labs exposing different parts of the body and getting effects in completely different regions, right? So I don't have to shine stuff in your eye. I don't have to do something which you're going to find uncomfortable or awkward. We just need to expose one part of your body, and if we wait 24 hours, we'll get it to kick in.
Wow. I wonder what the minimum effective dose is, because the back's quite a big area, right? Like I guess my question is, how much could you shrink that down? What's the threshold where you could shrink it down to actually get the same effect?
Okay, really good question.
Would that be...
Well, when you think about retina, when I'm doing the eye, I mean, that's an area like that. That's pretty small, but it shouts. It shouts at the rest of the body. I think we're probably talking about maybe 10 square centimeters. It may be the case that if we cut it down further, we get an effect, but maybe the effect takes a little bit longer to kick off. That's quite possible. There's so many things here. We know it works.
Wow.
We know that the mitochondria have got their own clocks. The mitochondria kick themselves into action. I think it's related to perceived dawn, and they kick themselves into action about half an hour before perceived dawn. And then when they're working really hard, you can give them red light and they'll work even harder. If you catch them at 3 o'clock in the afternoon, you can give them buckets of red light and nothing happens. And that's true in a fly, and it's true in a human being. And then I'm starting to get contacted by people saying, my kid's got mitochondrial disease. They can't move very well. They can't open their eyes. What can I do about it? And I had to say, I don't have ethics for this one. It's children. This is a big issue. And they started treating with red light. And the results, they threw me into a spin. Some of these children within a week are opening their eyes properly. These children that are relatively immobile have got improved mobility. So we have got ethics for it now. We have got a clinical trial in place. We are treating kids with red light, and the effect is very impressive. Now, theoretically, it all makes sense. My lab said to me, of course it will work. Of course it will work. And I said, this is a big leap. This is a big leap. And it did work. The first child and the others are following in exactly the same time course, probably could walk 30 to 50 meters. That first child, she's now walking a couple of 100 meters to school, and she's having swimming lessons. Even if it was that one child, I'd be over the moon. But we've got a whole collection of them now slowly moving forward. They're all very variable. They're all very different. My first target was the ability to open their eyes. That's working. And then all these other things are coming along. Their mobility is improving. They very often, because they can't control their eye muscles, they've got double vision. These kids don't have double vision anymore. If they stop the red light, things drift back. But then we can always pick them up again. So, sometimes they get infections because they're complex. It's a complex disease process. And things then start falling apart. and then you bring them back with a red light, and they're okay.
What diseases?
Okay, we talked about energy density, we talked about power density, but let's talk about how often to do red light therapy. Should we do this dose of 25 or 33 once a day, every day? Is that our daily dose? Well, there's an important concept called the cumulative dose response. And here's one quote from an article that's very good. The dose from one treatment lasts some time, and what remains of the dose is added to the dose of the next treatment. Adequate time between doses is essential to allow the cells time to respond to the initial dose, and also to avoid a situation where the accumulated dose eventually ends up above the biostimulating range or even in the inhibitory range, with consequently poorer results. So if we look at this graph, a lot of the cellular effects we know happen after a red light therapy session has ended. Some of the studies say between 3 to 6 hours, you actually get a peak of ATP response and some of those molecular effects. But then it kind of drops back down. But if you do a repeated kind of daily dose, you could be starting at an elevated level. Now, this might be a good thing, because if you take your time and do low doses and you gradually build up, that's why there's kind of a delayed effect, that sometimes it takes a few doses or might take a few weeks for you to start to really feel the benefits of red light therapy. But we can see if we keep going at that pace and we keep stacking on these effects, you could actually reach an inhibitory region similar to the biphasic dose response of a single dose. Now we know if we keep stacking the doses, then you also get an inhibitory response. And I think the opposite is happening too. That if you start out with high doses, your first couple weeks or months could be great. They might be miraculous. But over time, you might notice the benefits are tapering off. Maybe they're diminishing. Maybe they're even getting worse. And what's your natural instinct when that starts to happen? You might actually try to increase your dose to offset it. When in reality, and what the science shows, is that you should be decreasing your dose to maintain those benefits. And that could be done by reducing the frequency or reducing the dose. So another article that became viral recently is the effect on blood glucose. That they treated people 45 low intensity are also important for the systemic effects of full body red light therapy, that we need to recognize it. It's not a direct treatment that you might use lower doses. It's more of a systemic treatment that you need longer exposure times. So regardless if it's 25 or 33 joules per centimeter squared, that might be an effective range, that the study got good results with this dose. So the question would be, should we increase the dose? Would we get a better response if we do more, if we do 40 or 50 joules per centimeter squared? Or should we decrease the dose? It depends on how your body responds to red light therapy. But we know in general, a lot of studies have referenced this biphasic dose response, this hormetic upside down U curve, that if you get too much of any pharmaceutical, any therapy, too much water, too much oxygen, it ends up being a bad thing. One quote in a paper by Dr. Hamblin is very nice, so I'll read that to you. The biphasic dose response describes a situation in which there is an optimum value of the dose of PBM, most often defined as the energy density joules per centimeter squared. It has been consistently found that when the dose of PBM is increased, a maximum response is reached at some value, and if the dose is increased beyond that maximal value, the response diminishes, disappears, and it is even possible that negative or inhibitory effects are produced at very high fluences. So we know if the study decided to use 25 or 33 joules per centimeter squared, maybe we could...
If the light source is in contact with the skin, a lot more light will go in.
I like
So
that.
I just find it ridiculous hanging your panel on the back of a door and standing in front of it.
Yeah, that's fine.
If you stand in front of an LED panel, a surprising amount of the light is diffusely reflected off your skin. If you lie on the LED panel, much more of it goes in. So a relatively brief exposure to light can affect cells and tissues for a long time, and even as long as a month. We have data in mice that a single exposure to light can last for a month. As I said, the two major effects of this switch from glycolysis to oxidative phosphorylation. The first one is activation of stem cells. Because stem cells live in a hypoxic niche, they carry out glycolysis because there's no oxygen. But when their mitochondria are activated, they leave their niche in search of oxygen, and that activates the proliferation and differentiation programs in the stem cells. So it really mobilizes the stem cells from their niche, and quite often that's in the bone marrow. The other consequence of this switch from glycolysis, it switches the microglial phenotype from M1 to M2. And why is that important? For many brain diseases, and Alzheimer's is a principal one, but traumatic brain injury, stroke, depression is characterized by neuroinflammation. So your microglia in the brain are polarized towards M1, which means they're churning out pro-inflammatory mediators, but more importantly, they can't carry out the function of getting rid of the rubbish. So the microglia that get rid of the rubbish are M2, and they're anti-inflammatory, and they phagocytose the amyloid plaque and the aggregated synuclein and all the other protein rubbish you accumulate in your brain. So the photobiomodulation has two effects. It stops them churning out nasty inflammatory mediators, and it helps the garbage disposal activity. Inflammatory, the antioxidants, pro-survival, angiogenesis, lymphatic drainage, increased blood flow. So the beneficial effect in the brain, multifactorial. And for any given brain condition, it's not immediately obvious which of these is predominant. I mean, they're all good, but you could say, okay, for brain injuries, clearly repair is important. But, for neurodegeneration, for Alzheimer's and Parkinson's, helping the brain to repair itself could be the critical thing. Because you're not really going to...
Military looking into that now, in addition to the traumatic brain injury, that should be an important...
Right, and there's a big overlap between chronic TPI and PTSD and depression, anxiety. They all mingle together, It's almost impossible to treat one of these disorders on its own.
One of our speakers she's going to be speaking exactly about that. She deals with concussion all the way through to the senior dementias, and they're all just tied together, in her opinion. All right.
You notice with brain photobiomodulation is everybody sleeps better, basically. So this was Fred Schiffer, who some time ago we did this study for depression and anxiety. It was a single exposure to the head with Hamilton Depression and Anxiety Scale 2 and 4 weeks after a single treatment with this little near-infrared LED device on the head. And what you find is that 2 weeks after the single treatment, virtually everybody improved with both the depression and the anxiety scale. What you see that interfacial water in the mitochondria, its viscosity is lowered when it absorbs red near-infrared light. And because ATP synthase, unit 5, has a little molecular rotor, if you reduce the viscosity of the water, it'll turn faster. So folks like Andrew Sommer in Germany believes that's one reason why you get extra ATP with photobiomodulation.
Cells love to be together with their lymphatic vascular system when they're at the resting phase. So now we're going to take a virtual tour as if we have a special glasses that allows us to dive into our skin. What you see in green are the stem cells, in red are the blood vessels, and in white you can see our lymphatic vascular system. So now we're going to take this virtual tour into our skin, and what we found is that the lymphatic capillaries create protrusions that emerge from them and hold the stem cells tight when the stem cells are young and in the resting state. So you can see it also here. Essentially, what we found is that the communication between stem cells and lymphatics is controlled by the stem cells themselves. So the stem cells kind of like have the brain to control how their environment will look like. They will secrete factors to control the association with lymphatics when they're at the resting phase. And as soon as they will become activated, they will switch to a different program to tell the lymphatics to stay away for a little bit, allowing tissue regeneration to happen. And this is what happened throughout the cycle. So it's important for the stem cells to control how lymphatic behavior will look like. So the environment is important to maintain balanced stem cell fate decision. But the question remains, why hair loss happens when we age? Are we losing the stem cells? Are they just gone and not growing hair anymore? Or are we losing the soil, the environment, the niche that support the stem cells themselves? What are we losing when stem cells age? And this is a very complex question to ask because our tissues are composed of so many stem cells, so many cell types, so many cues and signals. Imagine that when lymphatics are no longer functional, when they're no longer able to make the association with their stem cells no longer synchronized. You can see some small stem cells here in green and some very big follicle together with them. And when that happens, tissue regeneration doesn't happen anymore because synchronized behavior, that stem cells will work with their environment in a synchronized way, is important for repair and for the when we have wounds, so it will restore itself. Those big follicles sometimes even develop hyperplasia. So balanced stem cell fate decision didn't happen anymore. What happened to these mice that had dysfunctional lymphatics? What you're looking right now are basically two identical twins. They were born to the same mom at the same time and at the same place. One of these mice had genetic mutation that caused its lymphatics not to function as good, not to form those connections that I showed you with the stem cells. What we found is that these mice, even though they were young, they started to lose hair prematurely. They basically express signals, or their phenotype was kind of premature aging. So we can push aging fast forward, but